I wonder why the celery looks the way it does.
The celery looks the way it does through physical proteins. Proteins are created through a process called transcription and translation. This process turns DNA genes inside the nucleus of a celery into physical protein that you can see.
How does it do this?
Before we talk about how this happens, it is important to understand what DNA is. DNA is deoxyribonucleic acid, a self-replicating material present in nearly all living organisms. It is the carrier of genetic information. DNA is made up of molecules called nucleotides. Each nucleotide contains a phosphate group, a sugar group, and a nitrogen base. The four types of nitrogen bases are adenine, thymine, guanine, and cytosine. The order of these bases is what determines DNA's instructions or genetic code. DNA never leaves the nucleus of its cell, so the information on it must be copied in order for it to replicate. The copying process is called transcription, and the copy of the DNA is called mRNA, or messenger RNA. To make the copy of the DNA, an enzyme called RNA polymerase does many things in order for the RNA to be created. It binds to a specific part of the DNA, which is called the promoter, at the beginning of a strand at the Tata box. It starts to unzip the double helix. As the RNA polymerase continues to move along, it rezips the double helix behind it after it is finished copying. It uses one of the strands as DNA as a template for when it is copying. It matches the new nucleotides with their pair. A always pairs with U and G always pairs with C. This is the copy. Once it hits ' termination signal, RNA polymerase pulls off and unattaches from the DNA strand. Some finishing touches before it is completely finished. At the 5' end of the DNA strand, a guanine cap is added on the end of the strand, and on the 3' end of the strand, adenine is added as a cap. Caps make it easier for the mRNA polymerase to leave the nucleus and to protect it. Finally, before exiting the nucleus, a process called RNA splicing occurs to basically get rid of any extra or non-needed information.
Well, how does translation work?
Well, once you have completed the first step of transcription, you go to translation. In the process of translation, a structure called ribosome, a mix of proteins and rRNA, reads the information from a molecule called a messenger RNA, or mRNA, and uses this information to build a protein. mRNA slides through ribosomes, which reads the mRNA 3 letters at a time, each set called a triple codon. Translation is happening constantly in a normal bacterial cell, just like it is in most of the cells of your body, and it's key to keeping you healthy and alive. In translation, the codons of the mRNA are read in order, from the 5' end to the 3' end, or upstream, by molecules called transfer RNAs, or tRNAs. So basically, translation involves decoding a messenger RNA, or mRNA, and using its information to build a polypeptide, which is basically a protein, or chain of amino acids.